So we know that we have neurons in our nervous system, but we also have all these helper cells, all these other types of cells that do specific things within our nervous system and help our neurons work as efficiently as they can. And these are called neuroglial cells. And there are about 1 trillion neurons in our nervous system. And the neuroglia cells outnumber the neurons by as much as 50 to 1. So they are definitely, there are way more in number than the actual neurons, right? But maybe we don't know that much about them, or you don't maybe. So we are going to talk about them today. So the neuroglia, or glial cells, basically support and protect the neurons. They bind neurons together and form framework for nervous tissue. In the fetus, they guide migrating neurons to their destination. If the mature neuron is not in synaptic contact with another neuron, it is covered by these glial cells, and this prevents the neurons from touching one another and gives precision to the conduction pathways. There are 6 types of neuroglial cells. 4 types occur only in the central nervous system. The first one is what we call an oligodendrocyte. It's a big word. And they actually form the myelin sheaths that we find in the central nervous system. Each arm-like process wraps around a nerve fiber, forming an insulating layer that helps speed up the signal conduction. There are epithelial cells. These line the internal cavities of our brain. They are cuboidal epithelium with cilia on an apical surface. They secrete and circulate our cerebral spinal fluid. And this is the clear liquid that basically bathes our central nervous system. And then there are microglia. These are small, wandering macrophages that are formed white blood cells. And then there are microglia. These are small, wandering macrophages formed white blood cells Then there are microglia, and these are small wandering macrophages. They are thought to perform a complete checkup of the brain tissue several times a day. They wander in search of cellular debris in order to phagocytize it and basically keep the brain clean. The last type of the 4 are the astrocytes, and these are the most abundant glial cell in the central nervous system. They cover the entire brain surface and most non-synaptic regions of the neurons in the gray matter of the central nervous system. And they have lots of functions. They form a support framework of the nervous tissue. They have extensions that contact blood capillaries that stimulate them to form a tight seal called the blood-brain barrier. They convert blood glucose to lactate and supply this to the neurons for nourishment. Nerve growth factors are secreted by astrocytes that promote neuron growth and synapse formation. They have the ability to communicate electrically with neurons, and they may influence synaptic signaling. They regulate chemical composition of tissue fluid by absorbing excess neurotransmitters and ions. And astrocytosis or sclerosis is when a neuron is damaged. Astrocytes form a hardened scar tissue and fill space formerly occupied by the neuron. Then there are 2 types that occur only in the PNS, one of them being the Schwann cell. These Schwann cells basically envelop nerves fibers in the PNS. They wind repeatedly around the nerve fiber. This produces a myelin sheath similar to the ones produced by the oligodendrocytes in the central nervous system. They assist in the regeneration of damaged fibers. The second type is a satellite cell. They surround the neurosomas in the ganglia of the PNS. They provide electrical insulation around the soma, and they also regulate the chemical environment of the neurons. So those are the 6 types of neuroglial cells. 4 are found only in the central nervous system, and these 2 occur only in the peripheral nervous system. Glial cells and brain tumors. Tumors are masses of rapidly dividing cells. Mature neurons have little or no capacity for mitosis and seldom form tumors. But brain tumors can arise from meninges, which are the protective membranes of the CNS, by metastasis from non-neural tumors in other organs, meaning something has broke loose from another tumor in another part of the body and has metastasized or moved. But most of them come from glial cells that are mitotically active throughout life. Gliomas grow rapidly and are highly malignant. And depending on the type of treatment that you may need, if you have a brain tumor, you may have chemotherapy, or you may have radiation or surgery. The brain is obviously a tricky one because of the blood-brain barrier. It can decrease the effectiveness of chemotherapy because it kind of watches what gets in and out. Myelin. What is the myelin sheath? Well, it is an insulating layer around a nerve fiber. In the CNS, it is formed by the oligodendrocytes, and in the PNS, it is formed by the Schwann cells. It consists of the plasma membrane of glial cells, 20% protein and 80% lipid. The term myelination basically refers to the production of the myelin sheath. This begins at about the 14th week of fetal development. It proceeds rapidly during infancy, and it is completed in late adolescence. Dietary fat is important to the nervous system development, and that is because, as we see, 80% of the Schwann cells or the oligodendrocytes is composed of 80% of lipids. So that high fat diet can be very beneficial for this myelin sheath to develop. And you want your cells to contain myelin. You want this act of myelination to take process, and you want it to be completed correctly. So in the PNS, Schwann cells spirals repeatedly around a single nerve fiber. In the CNS, oligodendrocytes reach out to myelinate several different nerve fibers in their immediate vicinity. In the PNS, basically, these Schwann cell spirals can lay down as many as 100 layers on their own membrane. There is no cytoplasm between these membranes. There is a neurolemma, which is the thick outermost coil of the myelin sheath. And this contains the nucleus and most of the cytoplasm. External to the neurolemma is the basal lamina and a thin layer of fibrous connective tissue called the endoneurium. Then we have the CNS. The oligodendrocytes here basically are anchored to multiple nerve fibers. They cannot migrate around any one of them like a Schwann cell. They must push newer cells of myelin under the old ones. So myelination spirals inward toward the nerve fiber. Nerve fibers in the CNS have no neurolemma or endoneurium. Many Schwann cells or oligodendrocytes are needed to cover one nerve fiber. The myelin sheath is segmented. There are gaps between these segments, and they are called nodes of Ranvier. There are also internodes, and this is where the myelin covered segments from one gap to the next. The initial segment is the short section of the nerve fiber between the axon hillock and the first glial cell. And the trigger zone is the axon hillock and the initial segment. And this plays an important role in initiating a nerve signal. So here is a picture with all the things that I just described. So basically you have the nerve in the middle, and you have this fatty substance that has formed around the nerve. That is the myelin. This is an example in the PNS. You want these myelin sheaths because what we're going to find out is that your nerve impulses, the stimuli, jump from node to node. So when you have a myelinated nerve fiber, it conducts these signals much quicker than a non-myelinated nerve fiber. It has to do with the rate at which these signals can move, meaning how fast you can respond. Now, there are some things that sometimes our body goes through that destroys our myelin. One of them is multiple sclerosis. If you've ever heard of multiple sclerosis, you may know it has to do basically with the loss of your muscles. And that is because it is an autoimmune disorder where your own body starts to attack its myelin sheaths, and it causes these myelin sheaths to deteriorate. And then the myelin is being replaced by a hardened scar tissue. So the nerve cell is disrupted. So it can cause double vision or tremors, numbness, speech problems, all kinds of things. And that is because it's just slowly basically disrupting the communication of your nervous system. Instead of the effectiveness that we want in our nervous system, where we have a signal, basically, like I said, jumping from all these myelin sheaths to the next myelin sheath to the next myelin sheath and performing a very quick, efficient signal, we have this disruption. So then maybe the muscle doesn't get the right signal and you have a jerky reaction, or maybe no control at all. The onset of this disease is usually between 20 and 40, and it can be fatal in the end. There is not a cure, and we're not 100% sure, and when I say we, not myself personally, but science isn't 100% sure really where this comes from or why all of a sudden we start to see it come to, fruition in a person. There's also something called Tay-Sachs disease, and this is a hereditary disorder of infants that we see usually in people of Eastern European or Jewish ancestry. And this is an abnormal accumulation of a glycolipid called GM2, within the myelin sheath. This is normally decomposed by a lysosomal enzyme, but the enzyme is actually missing in people that are homozygous, meaning they have two alleles for this Tay-Sachs. So instead of having the enzyme that would normally break this down, you see an accumulation of it, and that is a problem. This accumulation disrupts the conduction of the nerve signals, just like we saw in the multiple sclerosis where the myelin is getting deteriorated. This can cause blindness, loss of coordination, and dementia. And if you get it as a very little person before the age of 4, it can even be fatal because your nervous system is still developing. All right. Well, that was a lot of information, but really interesting, I hope. So please forward any questions to me and take care.